All right. So, hi everyone. It's really good to see a lot of familiar faces and really exciting to see a lot of new ones as well. Um, if you have questions about this stuff, I know everyone's tired, but I'm happy to talk about this stuff afterwards as well, if you have questions. With that being said, I'd like to share some of the things I've been working on over the past several years with a little bit of a gap in between. Um, Now, I'm not going to go over too much of this because Philip Cameron did an excellent job of explaining the relationship between structure and function of aptamers, why it's important, why it's useful. Uh, but in just a short brief, it really helps guide our intentional design elements that we can include on our aptamers to optimize them for better function. Helps us to sort of predict what kind of side quests our aptamers might go on with these kinds of optimizations, um, and help us improve our prediction power when it comes to computational studies on aptamers such as this. And I have an example of the RKEC one that Cameron showed earlier when he presented. So we decided to look at this structure-function relationship through this serotonin-binding aptamer. Now, a meta-analysis performed in 2022 found that about 13% of adults in the United States take antidepressants. Of these, SSRIs, or selective serotonin reuptake inhibitors, were the majority of those that were prescribed. However, misuse of this drug often leads to something called serotonin syndrome, which has some very nasty side effects, which include things like increased heart rate, delirium, and a host of other, oops, where's my, there we go. There we go. And a host of other issues that are very, very bad. So a system that can monitor these people who struggle with serotonin regulation in real time would benefit greatly from something that can help them monitor serotonin levels in real time. So we specifically started by looking at this serotonin aptamer that was selected in 2018 by Milan Stojanovic. This is also the same paper that we got the dopamine aptamer from that we looked at. When he published the paper, he also published some CD studies that he had done on this aptamer, which showed that it formed a G quadruplex in the presence of ligand. Now, we also looked at the dopamine aptamer, which showed a G quadruplex, but once we figured out the structure, it seemed to be that that's not the case. But at the time, we didn't know this, and therefore we did some additional studies to try and confirm the presence of this G quadruplex. So a lot of the results I'm going to share today are going to be NMR based. The afternoon section is going to be all about NMR. This is great. So you saw this slide earlier today with Philip, but just to reiterate, we look a lot at the immunoregion that is down here, that I've blown up here. And we could look at individual hydrogens that are base paired together to make some conclusions about what kind of base pairs are forming, the lengths of stems, the loop regions, and stuff of that nature with and without ligand present. I've also highlighted the exact hydrogens that usually generate these signals in the GC and AT Watson-Crick base pairs. We can also use NMR to track changes as a function of ligand content. So we can look at individual peaks start to come in with the addition of, here in this case, cocaine to the MN19 aptamer. And we can see that structural changes take place once this aptamer is introduced to its ligand. And we can use that as well to make some conclusions and gauge what kind of structural changes take place with ligand binding. So that sort of is what I'm going to be talking about for the majority of my presentation. So with that serotonin aptamer, the first thing we looked at was the 1D NMR. At the very bottom here, I have the free state of this aptamer. Even without the presence of ligand, without the presence of serotonin, it seems to form some structure. But what was weird was when I added the serotonin ligand, we didn't really see many changes take place, even when I went to a 1.3 ratio of serotonin to aptamer. So I stored away the sample in the fridge for several weeks and worked on other things. And Philip suggested that perhaps this aptamer just needs a little encouragement. Try throwing some potassium chloride in there, see if that helps it fold up. So I took the sample out 24-ish days later, and before I added the KCL, I ran it in the NMR one more time to see if anything had changed. And lo and behold, it looked completely different from what it looked like the day I had ran it, which sort of raised the question about whether or not this slow structural change implied slow binding as well. So in order to, I guess, take a look at whether or not that was the case, some of my mentors, Sljana Slakovic and Aaron Shora, did some experiments as well. The ITC performed on the left by Sljana found that there was no real evidence to show this slow ligand binding. Every injection here was about 300 seconds in between each injection. All the peaks were nice and sharp. She got a KD of about 0.24 micromolar. No issues there. Aaron had performed some fluorescence studies using the intrinsic fluorescence properties of serotonin. And again, nothing really out of the ordinary with regards to binding. So it seemed like these structural changes that took place over a very long period of time were just that. The binding actually took place relatively quickly in the normal time frame that most aptamers bind their small ligands. So I went back and performed a time course experiment 3 years later. I just took I started with the free state at the very bottom. I added some ligand and just called that time equals 0. And then I took a 1D spectra every half hour. I didn't include them all here because they would all just look like straight lines because of how smooshed they'd be, but I just added a few to sort of drive home the point. It seems that this aptamer starts to see these new peaks come in at around the 2-hour mark. And by the 8 to 10-hour mark is really when this aptamer finds itself in this final sort of folded, stable structure, which is very, very odd. And over the course of my entire time working in Philip's lab, I've looked at maybe 36, 37 different aptamer ligand systems, none of which have shown this very interesting and odd characteristic of slow binding. And just, I guess I didn't write it here, but this was also at room temperature. Normally, when we do these kinds of NMR studies, they're at 5 degrees to try and help the aminoprotons signals sort of come through a little easier, but these were all performed at room temperature. And just to sort of, I guess, pinpoint the change over time, I took one of the more resolved peaks, so this one right here, and I just plotted the intensity as a function of time. It doesn't seem like it was done even after 12 hours, but that's unfortunately as long as I had the NMR booked for at the time. If I wanted to redo it, I would have to book it for maybe 15 hours, maybe. But nonetheless, what we can gather from this is that by the 8 to 10-hour mark, most of it has already folded. I would imagine that if I'd gone for another 3 or 4 hours, this would plateau about here. But that's just me speculating, and I don't really know what I would do with the information if I did have another 4 hours of data here. But nonetheless, very odd behavior of this aptamer. Doesn't really make it very ideal for assays that require rapid structure change to generate responses. So we moved on. Fortunately, Cassie from the University of Columbia was very kind. She shared with us a serotonin-binding aptamer sequence, which is based on the original on the left, with some circular permutations made and some minimizations. So this is a much smaller aptamer. I think it's 32 bases. And it retains those G-rich regions. So we suspect this aptamer still likely forms a G quadruplex. But again, we're working with the new sequence. There were some modifications made. It's best if we double check that this aptamer still forms the same structure. So first thing I did was throw it in the NMR. And unlike the original, there isn't much structure present in the free state, which makes sense. We've shortened it. We've switched around the 5 and 3' ends. It's not terribly out of the realm of possibility that we have less structure formed in the free state, but we do see structures start to come in very nicely as we add in serotonin. Unlike the original, this all happened fairly rapidly. I did not have to wait 24 hours for this thing to fold up. Interestingly, once I got to the 1-to-1 ratio of aptamer to serotonin, we have this nice folded structure. But upon adding 10 equivalences of KCL, we see some additional structure changes take place. Some peaks separate into smaller peaks, and others converge into larger peaks. So it seems that there are some additional structure changes that take place with the addition of KCl. So I did the reverse. I started with the free aptamer once again, and instead titrated in KCl. And when I got to that 1:10 ratio, it seems that this aptamer can fold with just KCl. But in order to form that final stable bound structure, it requires serotonin. And then we can get that same spectra that I got in the last slide. So KCl alone may be able to encourage the formation of a G quadruplex, but really to get that full bound structure, we need both of them present. This is just a summary of the two slides I showed earlier. This is the free state at the bottom, just aptamer, no KCl, no serotonin. With just serotonin, it's mostly folded, no problem. And upon adding KCl, we see some slight changes. With just KCl and no serotonin, there's a lot of low intensity peaks present, but they're there. There are changes that take place with the addition of KCl. But again, to get that final form, we definitely need to add serotonin. And again, no evidence of that slow structure change taking place over multiple hours. So in order to confirm the presence of a G quadruplex, we took some inspiration from Professor Mart's lab, and we decided to perform an NMM binding study. So NMM is N-methyl-mesoporphyrin 9, which selectively binds G quadruplexes and then fluoresces as a result. Unlike most other porphyrins, it's very soluble, so easy to use. Because of the two propionic acid groups at the end here, so easily soluble in water, very easy to do very quick fluorescence experiments with this. This was no problem at all, and it does not have to be covalently linked to anything. So when I looked at this aptamer using NMM, I ran 6 different samples. The very top left is just free NMM, and I excited this sample at 399 nanometers and collected the emission spectrum from 600 to 650 nanometers. That's typically where we'd expect the emission to occur for NMM when it fluoresces. We saw no such emission with just free NMM, which is good. It shouldn't fluoresce on its own. That would kill the point. With just free aptamer, again, we saw no fluorescence, no emission here, so good. Just in case KCl for some reason caused it to fluoresce, I also ran that with NMM. No changes, very good. NMM with the serotonin was a question that may come up. Again, no fluorescence, so far so good. However, when I got to the combination of serotonin with the aptamer, which should, in theory, form G quadruplex when they bind, we in fact do see the fluorescence takes place, and the same goes for when KCl was added as well. So it seems that this aptamer does indeed still form a G quadruplex despite the changes and modifications that were made to it. So in order to further probe the G quadruplex that seems to be present, we decided to make some mutations to it. We picked out the Gs that look the best and partially arbitrary, partially, educated guess. But we took 4 different Gs and replaced them with As to see how that might affect their function when it comes to binding serotonin. Assuming that we've disrupted G quadruplex, we would likely see no binding, and if it doesn't seem to affect the binding, we would make the conclusion that this G might not be part of the G quadruplex. So looking at the first variant, which we've named V1, which is this G5 to A variation, starting at the very bottom. Interestingly enough, this variant that had the G to A replacement seems to show more structure in the free state compared to the SRTN12. But nonetheless, when I titrated in serotonin, we didn't really see much change takes place. Some signal intensity is lost here. But once I've gotten to the 1:5 ratio of aptamer to serotonin, I added 10 equivalences of KCl, and suddenly this aptamer folds up quite nicely. So it seems by changing this G to an A, the 5th position G to an A, we've hindered the ability for this aptamer to form a G quadruplex, but not eliminated it. And now it just needs potassium to encourage it to form this G quadruplex. and likely bind serotonin. V2, which was a G19 to A change, again, showed very similar structure to V1 in the free state. And again, similar to V1, no real significant changes took place with the addition of serotonin, even at a 1:5 ratio. And even adding 10 equivalents of KCl did not change that. I just have lots of loss of signal, but really no new peaks coming in and nothing that resembles anything like the original or V1. We made the conclusion that this is likely not binding, and we've likely disrupted the formation of the G quadruplex. So G19, very good likelihood that it is involved in forming the G quadruplex. V3 was the G23 to A change that we made. Slightly different structure in the free state compared to V1 and 2. But again, similar to V2, no real changes that take place with KCl and serotonin addition. So we made the conclusion that this aptamer or this G23 is probably important for binding and forming the G quadruplex. And finally, we looked at a fourth variant, which replaced G30 with A. This one was a little interesting in that it seems like it's doing its best to bind, especially when I get to the 1 to 2 and 1 to 5 equivalents of aptamer to serotonin. There are some changes that take place, but it's especially noticeable when I add the KCl. There are a lot of these little peaks that seem to be coming in. So it's possible, at least this is my running hypothesis at the moment, is that by replacing G30 with an A, we've significantly reduced the ability for this aptamer to form a G quadruplex, but the neighboring Gs are attempting to substitute themselves, but it's not the optimal geometry for this. So it takes a lot of encouragement for them to try and form the G quadruplex. But again, this is just my guess at this moment. I have no evidence to back this up. Yeah, so this brings me to my future steps. There are many to list, but I'm only going to list the most important ones. The qualitative data of NMR is great, but it would be great to have, even better to have some ITC data to sort of quantify how well or poorly these variants are binding serotonin, as well as some other variants we may or may not look at to probe this quadruplex even further. We could also perform, or I would like to perform, some NMM binding studies on the variants, as I did with the original version, to again have a second method to show that this quadruplex is not forming, other than be saying that, look, no peaks, there's no quadruplex. Finally, depending on how far we get with the additional NMR and the NMM binding studies to sort of probe the G quadruplex, we might potentially look to assign the NMR for this minimal serotonin binding aptamer. With that being said, I'd like to thank my lab mates, some who are present here, some who are not, the organizers for the event, as well as New Ventures for sponsoring part of my trip here. And with that, I will be happy to take any questions.